So, welcome everyone to our webinar today. We will be breaking barriers in plant spatial biology with some very exciting research, amazing images. I had a preview and amazing guests. So, for starters, I would like to introduce myself and my colleagues from 10x Genomics and Millennium Science. Millennium Science is a distributor of 10x Genomics across Australia and New Zealand. And my work focuses on supporting researchers in starting that journey. Today with me, I have also Jeff McDermott, Science and Technology Advisor from 10x Genomics, who knows a lot about 10x. And recently, I'm very happy to introduce Lim-Chi Liu, who is our plant expert and 10x expert as well. She recently joined Millennium Science, and I will let you, Lim-Chi, introduce our guests.
So Xiujuan is from University of Adelaide under Professor Matthew Tucker's lab. Today she's going to share with us how she used Visium spatial transcriptomics to study grain filling in barley. And Xiujuan, there you go.
Thank you, Ling-Chi. Good afternoon, everyone. Nice to meet you online. Thank you for your introduction, Ling-Chi. I'm from Weight Research Institute, University of Adelaide. My research interest is reproductive development in cereal crops, especially the ovule and the seed development. Today, I would like to highlight how the cutting-edge spatial transcriptome helps me understand the maternal control during grain filling in cereals. Cereal grains lies at the heart of the whole industry, not only because the grain can be directly consumed as staples for human and food for animals, but also as the primary materials in brewing industry or biofuel production, but also because most of the breeding efforts, such as optimizing the tissue architecture, improve the nitrogen utilization efficiency, or guarding the germ cell fertility, are aiming to improve grain yield potential and grain nutrient quality. So here we use barley as a model plant of cereals because it's one of the most important crops in Australia, and also because barley has a relatively simpler genome compared to wheat, of course. So barley grain is actually a fruit. It's a fruit called Caryopsis containing one single seed. Now I'll change to the pointer here. So the barley grain is developed from a fertilized pistil, which you can see from here. This is day 2, then day 5, 7, 9, 10, and then about 20 days. In details, this is what inside of the pistil. You see a single ovule here. The germline here within the ovule will become endosperm and embryo after fertilization, while its surrounding maternal tissue will carry on and become part of the grain to support the seed development. For example, the ovule wall will become the pericarp, integuments and the nucellar epidermis will become seed coat, and the nucellus itself will become an important transfer tissue called the nucellar projection to support the grain filling, which is we often call a grain filling because the grain is like a pocket. You fill it to full. It actually indicates the growth of the endosperm. So this is exactly how the maternal tissue supports endosperm growth. The nutrient supplies are from the mother plant is transported along the rachis of the spike and reach the vascular bundle embedded in the ventral part of the pericarp. After unloaded from the vascular bundles, the maternal supply will pass through the nucellar projection, then released to the cavity between the maternal and the filial tissue, then taken up by the endosperm transfer cells here and finally get into the endosperm for assimilation. Here I use a fluorescent tracer to visualize this. First, you see the green signals around the vascular bundles. Then it will pass through here to reach the nucellar projection and finally get into the endosperm. Please note that the endosperm here is coated by a cuticle layer, which is pretty much blocks the material exchange, leaving the only path for gray filling is via nucellar projection. So in a way, the nucellar projection is similar with the umbilical cord in human to link the mother and the child. So it is rational to speculate the nucellar projection is gating some way, is gating the maternal supplies provided to the endosperm. So previously we employed the laser capture microdissection, which can be considered as the last generation spatial transcriptome, and identified a transcription factor that is preferentially expressed in the nucellar projection. We temporarily named it as grain protein content 1 is because the mutant we created by CRISPR shows this almost doubled protein content in its endosperm without changing too much in the starch or beta-glucan content. Surely we're interested in investigate the molecular mechanism of how nucellar projection expressed the gene can affect the filial tissues. But the nucellar projection is the only small part of the whole grain. For example, this is a 2 weeks old barley grain. And then if you cut through the middle to see this section, here the orange part is the nucellar projection. It only occupies about 1% of the whole grain in terms of volume. And of course, you can expand how much noise we will have to deal with if we simply extract the total RNA in wild type and mutant for a transcriptome analysis. Of course, we can go back to laser capture microdissection, but it's really takes lots of efforts to do that. So the Visium actually provides a terrific solution for my challenge. So I performed the spatial transcriptome analysis in both wild type and the mutant. In terms of cell clustering and the grain entity, the mutant actually quite similar with the wild type. We also performed the traditional bulk RNA-seq in both maternal tissue and the filial tissue and identified around 17 to 1800 DEX respectively. Then we can actually locate those DEX in spatial atlas and sort them out in the tissue of interest. There are many surprising results actually from here. For example, here I showcase some DEX in maternal tissues. Some genes are upregulated, but it maintains a similar spatial pattern. Some genes are upregulated, but it expands its expression pattern by active the neighboring cells started to express this gene. And very rarely you see a completely changed spatial pattern for this gene. For example, in the wild type is expressed in the transfer cell, which I believe is a transfer cell, but in the mutant, the transfer cell stop expressing gene, but the outside epidermis start to express this gene. So it's that indicate that the from RNA-seq, you can only see a number like log2 fold change, but the actual spatial pattern can show much more diverse. Here are some DEX in filial tissue. Again, this gene is downregulated because those cells are just expressing less. This one is upregulated because it has expanded expression pattern. And again, sometimes the gene just start expressed in totally different tissues. So with this, we can actually have a better geo enrichment, which analysis you quite often see from the transcriptome analysis. But this time we can do the geo enrichment within the tissue context. For example, in the nucellar projection and vascular bundle, the upregulated DAX shows two keywords, transport and hormones, which is a little bit surprising. We didn't expect that. Here I showcase a sugar transporter and a negative component of GI signaling pathway. And in the endosperm, which the tissue where the transcription factor we care about is not expressed, the nutrients reservoir activities are actually heavily upregulated. It's a red big dot here. One of the gene is hoarding, which is a major storage protein synthesis gene in barley and sperm. And you can see in wild type, it's mainly expressed in the flanking endosperm. But in the mutant, it's ubiquitously expressed in the whole endosperm and even expand a little bit to the pericarp. This is probably can explain why the mutant accumulate more proteins compared to the wild type. But now we wonder, because this gene we care about, the GPC, is expressed in the nucellar projection, then we wonder if it's because in the mutant, the nucellar projection actually transport more building materials to the endosperm, for example, amino acids. So because it's a transcription factor, so we're adding another layer of proof. We use the cut and tag to explore the binding site of this transcription factor genome-wide and overlapped the target gene with Visium and with bulk RNA-seq. Then we narrow down the actual genes downstream of this transcription factor is about 100. So among those, we noticed several interesting transporters, including MFS, the lysine histidine transporter, and amino acid permease, which probably a universal transporter for amino acids. So those are interesting candidates for us to do a further validation. But this is not yet the whole story. If we look closer to the nucellar projection from the top to the bottom, 4 different types of cells can be observed. So type 1 cells are, they look round, meristematic. Type 2 cells are elongated and differentiated. Type 3 cells become round again, but because of the cell wall ingrowth, they look a little bit swollen. And the type 4 cells just undergo rapid cell death to release its cell content to the cavity for the endosperm transfer cell to uptake. So the UMAP actually suggests a very interesting result. So in the wild type, you can see this cluster is nucellar projection. It gives you almost a perfect linear pattern of those nucellar projection cells, echoing the 4 different types of cells in nucellar projection from the top to the bottom. While in the mutant, those dots looks less spreaded, indicating the change of cell properties in those subdomains. When we look at the sections of to observe the cell morphology, we notice that it seems the mutant actually generate more type 1 cells compared to the wild type. This also indicating that GPC1 not just regulating the transporting, but also regulate the cell patterning in the nucellar projection. Now we wonder if we can manipulate the gene expression specifically in nucellar projection to actually maternally control the gray filling. To achieve this, we need a tissue-specific promoter. So once again, with the help of Visium, we screened the cell cluster marker genes and find several candidates which are only expressed in the gray in a high level. And so this is one case, one example of the candidate promoter. With this, we can further do a tissue-specific overexpression, for example, to overexpress a amino acid permease, which I mentioned before, or even JAS, we can mess up with the hormone signaling pathway to see how the maternal tissue can affect the filial tissue. Now let's take a step back to look at the larger picture of gray filling. So as the last step of resource relocation from from source to sink, the nucellar projection and endosperm transfer cell actually form transfer cells actually form a maternal filial interface, which has been believed in many species to be the nexus of nutrient dynamics and seed development, despite of diverse cell types of structures in different plants, of course. For example, in Arabidopsis, TT16 is expressed in the maternal nucleus, the maternal nucellus, regulating the fatty acid content in the seed, while in maize, the sugar transporter sweet 4C is expressed in the basal transfer layer, regulating the seed filling. Now, when we look at barley, wheat, and oat, they use a very similar way to feed their grain through vascular bundle, then nucellar projection, then endosperm transfer cell. So we try to use barley here as a model to understand how maternal tissue interact with the filial tissue in this type of cereals. Now, with the spatial transcriptome, we can further explore the other side of this interface by looking at the transfer cell specific genes and pick interesting candidate for further functional validation. Additionally, we recently performed the Visium in oat grain as a part of the GRDC project aiming to optimize the oil content in oat. Even though I just mentioned that oat has a highly similar gray filling path compared with barley, the endosperm of oat actually has a different way to assimilate and to store its nutrients. That's why you don't see people pouring barley milk in their coffee. It will be a very interesting comparison in terms of understanding the difference between different major cereals, currently waiting for the sequencing results coming back. With this, I would like to thank my colleagues in Weight Research Institute and thank Marta. She's a pioneer for barley Visium from La Troupe, and also Limchi and her colleagues for the kind support. Thank you.
Thank you for this. This was really feast for my eyes. And I really appreciate let's clap. The comparison to actually like human development was really like helping me understand what's happening, with the plants. And I especially like the example that you showed, why you can't get this information from bulk RNA-seq because you will get the same value, but the distribution, like locally, it's like so different. It was fascinating. Just checking if we have any questions from our audience. Our audience is a bit shy today. Lim Chi, do you have any questions for now?
Yeah, I do have a question to ask. It's very nice to see that you start to apply that to other species like oat. Do you find any different in terms of applying the technology across different species?
Yes, actually, yes. So because I have experience in barley, we thought, oh, maybe we can skip the tissue optimizing steps in oat because they look similar. But we decide not because it's risky. It turns out the barley grain and oat grain have very different, let's say, nature or texture, because oat grain takes much shorter time for the permeabilization, like 10 minutes shorter. So that's actually quite surprising.
Yeah.
Okay. So for everyone's reference, don't skip that. Even though you see publications, don't skip the tissue optimizing.
Good tips, yeah.
Yeah.
So you highlight a few benefits as well. I can see that you say Visium is good for small population of cell, and also to compare between like specifically across for that
Yeah.
small population of cell between wild type and mutants.
Yes.
And then the third one, I think you mentioned is good to identify like cell type specific marker for downstream validations.
That's right. Because for like downstream validation, I want to do a tissue specific overexpression. I don't want to do a global overexpression, for example, use 35S or ubiquitin promoters, because that will cause lots of phenotypes that I don't want. So that actually helps a lot to narrow down the candidates.
Yeah, it's very nice to see how Visium help you for your research.
Question from Daniel. What would be the strategy to do the same across the whole grain filling period?
Well, you can still do a similar thing, but the stage I picked is 10 days, which is perfect for Visium because it's large enough. I mean, the sample size actually matters for Visium from my perspective. The younger grain is fascinating, but the grain development within 5 days after fertilization is currently a black box to us. We don't know what's happening there because the endosperm is liquid at that stage. So that gives much challenge for Visium. I know Marta has tried. And also for smaller tissue, you also have this challenge to get perfect angled of sections. Yes, that will be great to have a whole developmental stages, but for younger tissues, or younger stages, I think we probably need to combine with a single nucleus RNA-seq together to understand this better.
Hello everybody, my name's Jeff McDermott. I'm a Science and Technology Advisor with 10x Genomics, and I just want to start by saying thank you so much for being here with us today. We really do appreciate the time that you've taken out of your day. I just have some really quick slides to showcase the 10x Genomics product portfolio, which can of course be utilized within the plant and broader agricultural research. The slide just contains some important information about forward-looking statements that I may be making today. So to make any progress in unraveling the complexities of biology, you're going to need the right tools at your disposal. And you need technologies that provide insights at that fundamental resolution of biology, which is, of course, the resolution of a single cell. And it's that need that underpins the direction we take when building our products at 10x Genomics. And as you can see, we built 3 platforms that each allow you to take a look at single cell biology with a unique perspective. So we have the Chromium single cell, which, as the name suggests, enables high throughput single cell analysis, allowing you to profile transcriptome-wide gene expression and other molecular features at single cell resolution for deeper insights into cellular diversity and function. Next, we have Visium spatial transcriptomics, and this provides spatial whole transcriptome gene expression mapping at single cell resolution, allowing you to explore tissue architecture and study gene activity within the precise spatial context at tissue section level. And then finally, we have Visium in situ, which delivers in situ high-plex RNA analysis directly within the tissue sample itself, enabling subcellular localization and quantification of hundreds of targets for advanced cellular profiling and spatial biology studies. This slide, the numbers on here are a little out of date. They were put together in September 2024, but what you see here are plant-focused research papers that have utilized at least one of the 10x Genomics products that I just introduced. So as you can see here by this ever-growing body of work, all 3 of our 10x Genomics platforms can be used with plant material. Without diving too deep into the literature, I just want to showcase some common examples of why researchers have been utilizing the platforms from 10x Genomics in their plant studies. Shown in these next few slides are the main advantages of using single-cell and spatial transcriptomics in plant studies. I'll begin with studying cellular heterogeneity with precise resolution, and plants obviously have highly heterogeneous tissues where different cells within the same tissue or organ can behave differently depending on their location or developmental stage. So single-cell transcriptomics allows you to analyze the gene expression profile of individual cells, and by isolating and profiling individual cells, it becomes possible to identify rare or specialized cell types such as those involved in defense mechanisms or nutrient uptake that would otherwise be masked in bulk RNA sequencing data. You discover rare and novel cell types, and single-cell transcriptomics has led to the discovery of new cell types and states within plants that were previously uncharacterized. These may include intermediate cell states during differentiation or rare cell types involved in specific physiological processes. And then spatial transcriptomics can be used to complement single-cell findings by showing you where these novel cell types are located within any given one tissue. We're mapping gene expression in the tissue context. So speaking of spatial transcriptomics, this technique provides a 2D view of gene expression by maintaining the tissue architecture. It helps you as a scientist to understand how genes are expressed in specific spatial regions of plant organs, such as the leaf or root or stem, in relation to one another. This is important for studying how environmental signals like light and temperature and pathogens affect gene expression in different parts of the plant. Unraveling gene regulatory networks, and by analyzing gene expression at the single cell level, it becomes easier to construct these gene regulatory networks for specific cell types or regions within the tissue. These networks help you understand how different genes interact and control plant development and physiology, and this information is vital for identifying key regulators of important traits, such as disease, resistance, or growth rate and reproduction. We have understanding developmental processes, and plant development involves complex interactions between different cell types and layers within the plant tissue. So spatial transcriptomics allows for the mapping of gene expression patterns across the tissue whilst preserving their spatial arrangement. And it's really particularly useful for studying processes like root development, leaf differentiation, and floral organogenesis, where the position of a cell in relation to others is crucial for its function. Exploring plant environment and plant microbe interactions. Plants are highly responsive to environmental stimuli such as water availability, light intensity, and pathogens, as well as nutrient levels. So single cell and spatial transcriptomics allows you to understand how specific cell types or tissue regions respond to these external signals. For example, the response to drought stress in root cells can be profiled at much finer resolution, enabling identification of key regulatory genes and pathways involved in this stress tolerance. Gaining insights into plant reproduction, spatial and single-cell transcriptomics are particularly useful for studying reproductive organs, flowers and fruits and seeds, and that's because these tissues often contain highly specialized cells. And understanding the gene expression patterns in individual cells during key events like pollen development and fertilization and seed formation can provide insights into plant breeding and crop improvement strategies. And finally, improving crop traits with high throughput functional genomics. And both single cell and spatial transcriptomics can be scaled up to perform high throughput analyses of gene expression across multiple plant species, tissues, and conditions. And this provides a comprehensive view of plant gene function and its regulation across diverse biological contexts. So these technologies can be applied to crop species, enabling you to pinpoint the genes responsible for desirable traits, such as increased yield or stress resistance or even nutrient content. So hopefully it's clear just from those few snippets of data that single cell and spatial transcriptomics is transforming plant research by providing high resolution insights into the complexity of plant biology. These technologies allow you to explore the diversity of cell types, gene regulatory mechanisms, and environmental responses at unprecedented resolution, opening up new possibilities for understanding plant development, stress responses, and improving crop traits. So if you've been thinking about applying 10x Genomics single cell or spatial tools for your plant research, or you're truly inspired by what you see here today with our speakers, then certainly don't hesitate to reach out. We'd love to have a project discussion with you and see what's possible. So with that, again, I'll say thank you very much.